Dear students, we have studied the cell membrane, the structure, function, and also the cell wall and its function. Now in today's class, let us study the cell organelles. The living components of cell are called cell organelles. The cell organelles are found in cytoplasm of the cell. One of the cell organelle is endoplasmic reticulum. In today's class, let us study the endoplasmic reticulum. Okay. Endomembrane system. Each membranous cell organelle is distinct in terms of its structure and function. Considered together called endomembrane system because their functions are coordinated. The meaning of this sentence, if you analyze, the some cell organelles in eukaryotes are bounded by the membrane. The structure of the cell organelle and with respect to the function of the cell organelle together we call endomembrane system because their functions are coordinated. The endomembrane system includes or consists of endoplasmic reticulum, Golgi complex, also called Golgi apparatus, lysosomes, and vacuoles. All these comes under endomembrane system. Once again, endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles comes under endomembrane system. The cell also contains other cell organelles in the cytoplasm. For example, the mitochondria, the plastids or the chloroplast, peroxisomes. These are not coordinated in their function. And these are called or comes under non-endomembrane system. See, endomembrane system on endomembrane system. Mitochondria, chloroplast, peroxisomes. The reason is their functions are not coordinated. So that's all about endomembrane system. Okay. Now let us study the endoplasmic reticulum. The endoplasmic reticulum is a endomembrane system. The electron microscopic studies of eukaryotic cells shown the presence of network of reticulum of tubule structures distributed in the cytoplasm called endoplasmic reticulum. What is endoplasmic reticulum? The network of tubular structures present in the cytoplasm of the cell is called endoplasmic reticulum. Okay, it was discovered by Coleman. The endoplasmic reticulum divides the intracellular space. Intracellular space means the space present in the cell or within the cell into two compartments. That is luminal or inside the endoplasmic reticulum, and extraluminal, that is outside the tubes of endoplasmic reticulum, that is cytoplasm. So it is clear endoplasmic reticulum divides the intracellular space into two compartments or two parts, that is luminal and extraluminal, or you can say inside the tubes and outside the tubes. Outside the tubes means cytoplasm. Okay. Next, classification of endoplasmic reticulum. The endoplasmic reticulum is mainly classified into two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, in brief, RER, Why the endoplasmic reticulum is called rough? The endoplasmic reticulum on the outer surface of membranes are rough in nature because of the presence of ribosomes. Hence, it takes part in the protein synthesis. The rough endoplasmic reticulum is also called granular endoplasmic reticulum. It is double membranous system. It helps in the movement of materials within the cell. It also gives mechanical support within the cell or to this cell. It extends from nuclear membrane up to the cell membrane. That is rough endoplasmic reticulum. Smooth endoplasmic reticulum, which is also called a granular endoplasmic reticulum. What is this smooth endoplasmic reticulum? SER. Smooth endoplasmic reticulum in which the ribosomes are absent. If we examine the endoplasmic reticulum, in the rough endoplasmic reticulum, outside the cell membrane, ribosomes are present, hence called rough. But in smooth endoplasmic reticulum, the ribosomes are totally absent. So this is the difference because the ribosomes are absent. So this extend, that is double layer endoplasmic reticulum membrane, extend from nuclear membrane up to the cell membrane or plasma membrane. Okay. It occurs in three forms actually: cisternae, tubules, and vesicles. Okay. Next, function of endoplasmic reticulum. The rough endoplasmic reticulum is mainly take part in protein synthesis. Because of the presence of ribosomes on its outer surface, it takes part in protein synthesis and also secretion. Very important function of rough endoplasmic reticulum. Smooth endoplasmic reticulum. Smooth. As the name indicates, the surface is smooth because the ribosomes are absent. The smooth endoplasmic reticulum takes part in the synthesis of phospholipids and also glycolipids, that is phospho and glycolipids. Majority in the cell take place in the animal cell. In addition to this, steroidal hormone also synthesized. Okay. So now, this endoplasmic reticulum also take part in the formation of cell plate during cell division in plant cells. Okay. The function of rough endoplasmic reticulum and smooth endoplasmic reticulum, very important from the point of examination. Now study the diagram. See, at the center there is nucleus. Even the nuclear membrane is clearly visible. See the rough endoplasmic reticulum. You can see it is double layered membranous system. Inside this membrane there are channels. You can see the channels through which transportation of some molecules takes place from one region of the cell to the other region. Why this endoplasmic reticulum is called rough means you can see small dots on the outer surface of the endoplasmic reticulum. We call these dots the rough endoplasmic reticulum. Okay. You can also see the smooth endoplasmic reticulum. Just below the rough endoplasmic reticulum, the smooth endoplasmic reticulum, you can see. See the difference. Here there is no ribosomes. In smooth endoplasmic reticulum, the ribosomes are absent. A very important notable thing. Okay, you can see now the rough and smooth endoplasmic reticulum, the diagram. Okay, practice the diagram. Very important. In the examination, you are supposed to draw the diagram. You must practice the diagram. That's all about structure, types of endoplasmic reticulum, and also function of endoplasmic reticulum. Okay. Next, we shall see or study another cell organelle, Golgi apparatus. or Golgi bodies. Golgi apparatus. Sometimes we call Golgi bodies, Golgi complex. In the animal cell, we call dictyosome actually. The position, the number, and the size also varies. As the name indicates, the Golgi apparatus was discovered by Camillo Golgi. He observed the Golgi apparatus just near the nucleus or just above the nucleus. It is densely stained structure. It is a net-like structure we call reticular. Who discovered Golgi complex? Camillo Golgi discovered the Golgi apparatus. Okay. In a cell, usually or generally, only one Golgi apparatus is present in a cell. But if you examine the nerve cells, liver cells, and most of plant cells, the number varies. The number may be many. Okay. If we examine Golgi complex under electron microscope, it shows cisternae, small tubules and vesicles, large vacuoles. Okay. Cisternae. Golgi apparatus consists of many sac-like structures. Sac-like structure. These sac-like structures are called lamellae. They are flattened, double-layered membrane, and they are sac-like structures. The diameter of the cisternae varies from 0.5 microns to 1 micron in diameter. These cisternae are arranged parallel to each other. I told you the number, the shape sometimes varies. If you examine the cisternae, they are double-layered, flattened structures. They are sac-like structures. Sac-like means bag-like structures. Shows the forming phase. Forming phase means the convex phase. See the diagram, you'll come to know. And the concave phase. It is called maturing phase. Forming phase and maturing phase. Or you can say convex phase and concave phase. But interconnected. It shows small pores. Sometimes under the electron microscope we call fenestration. Okay. Intercisternal elements also present. Small tubules are also present. And in addition to that, small vesicles are also present. The tubules and vesicles develop from the lamellae. How? By budding or pinching. Pinching forms vesicles. At the edge, you can notice the presence of large vacuoles. Okay. So that's all about Golgi apparatus. The structure. See the diagram of Golgi complex. The cisterns are there. They are double layered membranous structures. You can see they are also interconnected. That's also important. Interconnected system. Okay. Now let us study the function of Golgi complex. Important for NEET or CET examination. The main or the principal function performs the function of packing materials either intracellular or extracellular. Another important function of Golgi apparatus. Golgi apparatus forms the site for the formation of glycoproteins and glycolipids. A very important point for NEET examination. Golgi apparatus or Golgi complex forms site for the formation of glycoproteins and glycolipids. Okay. In case of animal cells, if we examine the sperm of animal, above the sperm or the head, a cap-like structure is present called acrosome. The acrosome is derived from the Golgi apparatus. Another important function. In the animal cells or in the animals, it forms the site of for origin of skin pigment. Skin color is dark because of the presence of melanin pigment. Most important function of Golgi apparatus. Okay. It also takes part in the synthesis of or formation of cell plate during cell division in plant cells. That is cell wall formation in plant cells. That is also important. Okay. So that's all about Golgi complex or Golgi apparatus. So we have studied the endoplasmic reticulum, the structure and function. Then endomembrane system also studied. Then we have studied the Golgi apparatus and also structure of this. Function of endoplasmic reticulum. The function of endoplasmic reticulum and Golgi apparatus very important from need point of view and also for the main examination, that is annual examination. So that's all about endoplasmic reticulum and Golgi apparatus. Okay, now thank you.